Ladies and gentlemen, we are Ahili Tarapda, Julia Sunil, and Wong Intao from Raffles Girls School, and we'll be presenting our project exploring the role of Myristoylated Alanine-Rich C-Kinase substrate in colorectal cancer metastasis. This is what we'll be covering in our presentation. Let me begin with the key terms of our project. Colorectal cancer, or CRC, is cancer of the colon or rectum and accounts for almost 1 in every 4 cancer diagnoses worldwide. Despite its prevalence, however, a suitable biomarker has yet to be found for its metastasis. Myristoylated alanine-rich C-Kinase substrate, or MARCS, is a protein that has been established to promote metastasis of cancer in some parts of the digestive system, leading us to hypothesize that it may have a role in CRC metastasis as well. Thus, the aim of this project was to determine whether MARCS has any role in CRC metastasis, as well as to determine its potential as a possible biomarker for it. We aim to do this by observing the differential expression pattern of MARCS into isogenic, metastatic and non-metastatic cell lines, SW620 and SW480, as well as a non-isogenic metastatic cell line, HCT116. We hypothesized that the expression of MARCS would be higher in the metastatic cell lines, and therefore MARCS would have a role in the metastatics of CRC, as well as potential as a possible biomarker. The results showed that the expression of MARCS was indeed significantly higher in the metastatic cell samples, proving our hypothesis right. Our experimentation included cell lysis of the harvested samples, protein estimation to determine the volume of samples to be loaded, SES page to ensure similar loading of all samples, silver staining to develop the gel from SES page so as to make the loading visible, Western blot to detect MARCS and therefore determine its expression in the various samples, and finally colloidal silver stain to verify the results of SES page and silver staining.
The results of protein estimation. Deducting the average light absorbency value of the solution with no BSA from that of the other samples, the graph of light absorbency against protein concentration was plotted, and the function y equals to 0.0011x was derived, serving as a model for the relationship between the two variables. We had hypothesized that the greater the concentration of proteins, the higher the light absorbency values would be. This hypothesis followed true for all except for the last. The light absorbency value for 250 microgram per ml was lowest and was thus discounted. Using this model, we can hence estimate the protein concentration of the samples. The results of silver staining. The cell samples have been run in the following order of SW620, HCT116, SW480, followed by the marker. We had hypothesized that the loading, and therefore the total amount of bands, will be similar. However, the gel showed fainter bands for SW620 and HCT116. This could be either due to the fact that the loading was not similar, or that the gel had not developed well enough. Since the results of silver staining were ambiguous, a colloidal silver stain was conducted later. The results of Western blot. The all-blue marker is shown on the left, followed by SW480 and SW620 respectively. HCT116 proteins were not expressed. The pluses represent proteins of molecular masses 25 kDa and 75 kDa, moving up. MARCS has a molecular mass of around 80 kDa and is found right above the second plus shown on the marker. We had hypothesized that there will be a greater expression of MARCS in the metastatic cell lines SW620 and KHCT116 than in the non-metastatic SW480. After the MARCS markings were identified, it was observed that MARCS was expressed more in SW620 than in SW480. The results of colloidal silver stain. The PVDF membrane from the Western blot, after the colloidal staining, shows similar number of bands for SW620 and SW480. The bands for HCT116 did not develop.
From our results, we firstly concluded that the loading for all 3 samples was similar. However, the sample of HCT116 was not loaded properly, resulting in it not being expressed, and thus the results cannot be concluded from it. For this reason, this sample was discounted, and we drew our final conclusions based on the other 2 samples. It can be concluded that MARCS does have a role in CRC metastasis, as the expression of MARCS is significantly higher in metastatic cancer cells than in the primary cancer cells. If further research is done, MARCS could potentially be used as a biomarker for colorectal cancer metastasis. There are, however, some limitations in our experiment. As HCT116 was not expressed, our conclusion was based on only one sample of each metastatic and non-metastatic cancer cells. For more comprehensive results, more samples for each cell line and more different cell lines could be tested for more data to be collected. The experiment was also only conducted once, which greatly decreases the reliability of the results. Repeating the test more times would minimize human error and perhaps obtain an expression for the HCT116 cell line. Though it was concluded that MARCS has the potential to be used as a biomarker for CRC metastasis, the specific role it plays cannot be concluded from our study. Therefore, it cannot be established as a biomarker for CRC metastasis conclusively. Therefore, as a possible extension for our project, we could deepen our understanding on the specific role that MARCS plays in the process so as to assess its final suitability as a biomarker. We could also look into other possible biomarkers for CRC metastasis. From the results of the Western blot, other unidentified bands of proteins of various molecular mass that show difference in expression in the metastatic and non-metastatic cell lines can be observed. These proteins could be potential biomarkers for CRC metastasis. We could also explore MARCS as a biomarker for metastasis of other types of cancer. Since MARCS is known to have a role in the metastasis of cancer in areas such as the bile duct and now the colon, it could possibly have a role in cancer metastasis in other parts of the digestive system as well.